Hello everybody, I'm really happy to present our work entitled Past Environmental Changes Affected Lemur Population Dynamics Prior to Human Impact in Madagascar. So sorry, this work is not focusing in the central highland from Madagascar, but in northern Madagascar, but we think this is a really good example of research, multidisciplinary research, to try to answer to really complex questions, such as in central island of Madagascar. And this example might be applied more or less the same strategy in different regions, such as in the central Madagascar region. So first, I would like to focus about this research strategy and this project, which is a multidisciplinary project. And this project was led by two teams, by the team of Professor Utra Despil from the University of Medicine, focusing on demographic inferences on mouse lemurs and genomic data from this animal, and also with the team of Professor Herman Belling from the University of Göttingen, focusing on paleoecological record and paleoenvironmental changes. So then I worked with Helena Teixeira. She did her PhD with Utra Despil, and we worked together to reconstruct paleoclimate and paleoenvironmental changes in Montane Dam in northern Madagascar, and to try to compare and to validate demographic inferences made for one species of mouse lemurs from Montane Dam in the same region, and to combine these two datasets and to answer the question to see if this species was impacted by climate change and also human impact during the last millennia in this region. So to do that, we used a site, Montane Dam, in northern Madagascar, because this region, this mountain, is really a good area for paleoecologists to obtain sediment records, because there is many volcanic lakes which are particularly suitable for sediment coring and reconstruct past vegetation and climate dynamics during the past, and also because there is also a species, Microcebus arnoldi, which is dependent from this rainforest, and Microcebus are particularly suitable to reconstruct demographic inferences based on that species by using current genomic data of that species. So I will first talk about the paleoenvironmental changes reconstructed from the Lake Modi in the central part of Montane Dam, and then after that Helena will show her result about demographic modeling and how we compare these two datasets from the same site in Montane Dam. So this is the main vegetation data during the past 25,000 before present, and here are represented four main group of plants and pollen taxa, and I will just detail them now. So in green we have the evergreen humid forest, the rainforest that we have today in Montane Dam, mountain vegetation in brown, and two other taxa, which are the sum of aquatic plants in purple, such as, for example, Potamogeton, and in orange yellow we have the Poaceae, and in the black histogram we have microcharcoal particles representing the fire regime and fire activity in northern Madagascar surrounding Montane Dam. So what we can see is that before 15,000 years before present, we have a mixture of evergreen humid forest and mountain vegetation growing in Montane Dam, and showing certainly cooler conditions but also drier conditions at that time. And then for 15,000 years before present, we have an important shift about vegetation changes, showing that evergreen humid forest is increasing rapidly and mountain vegetation is decreasing also rapidly. And then this shows the beginning of the African humid period at 15,000 years before present, and then African humid period continue until 5,000 years before present. You can see that the rainforest is really stable at the beginning of the Holocene and remains stable after 5,000 years before present. However, we see that we have an important increase of aquatic plant after 5,000 years before present, showing certainly that aquatic plants are showing shallow lake condition. So then the lake level was certainly decreasing after the African humid period. However, this precipitation decrease explaining the lake level decrease was not strong enough, certainly, to change the rainforest growing in Montane Dam. And during the last millennium, we see an important decrease of rainforest at the expense of boissé, so grass, showing the last period of vegetation changes. And if we compare to fire regime or microcharcoal particles, so we can see that you have a short episode of fire occurring during the first phase of African humid period. Then we have almost nothing during the beginning of the Holocene until 5,000 years before present. You have just a few particles after 5,000, and then an important increase of fire and particles, microcharcoal particles, after 1,000 years before present, showing that fire were occurring on a regular basis surrounding the mountain dam. So we can also compare with sedimentological data made from the same core, and this data are showing increase of erosion at the beginning of the African humid period, certainly showing the increase of humidity. And then we reach maxima during the second phase of African humid period, and then after 5,000 years before present, we have a decrease of erosion and then precipitation on the site, and then this precipitation are still decreasing and erosion until almost today, based on our record. So with this picture here, we can resume the main result of past vegetation changes and climate dynamics. The first period, before 15,000 years before present, we had a mixture of mountain vegetation and evergreen humid forest under cooler and certainly drier also condition. And then after that, you have the second and the third period occurring during the African period, so an important increase of humidity. And during that period, we certainly had a maximum extension of the rainforest due to the maximum humidity of the second and the third period. So then, after 5,000 years before present, we have the end of the African humid period, the fourth period. The rainforest on the mountaintop was not so much affected, as we can see with our pollen record. However, the precipitation decrease was maybe reducing the rainforest distribution and affected maybe also the lowland elevation more than the rainforest in mountain dam and on the top of mountain dam. Then the last period, the fifth period, is occurring during the last millennium, and this is characterized mainly by an important decrease of fire activity, certainly made by human impact, and also climate condition, and mainly a precipitation decrease during that last millennium was also maybe affecting vegetation changes in the region at that time, in northern Madagascar. So now Helena will compare this period of paleo-environmental changes that we found with the sediment core from Lake Modi with demographic influence based on Microcebius arnoldi and model validation about that.
In this study, three complementary approaches were used to reconstruct demographic history of Microcebius arnoldi. The pairwise sequentially Markovian coalescent method, or PSMC, has been widely used to infer population size change from whole genome sequence. However, the PSMC estimates the inverse instantaneous coalescent rate, or IICR, of the ancestral population of a diploid individual, which interpretations are dependent on the model assumptions. Therefore, for a parametric population, the IICR corresponds to the trajectory of the effective population size over the time, while for a structural population with a constant population size, the IICR reflects change in migration rate between the populations. In addition to these two methods, the FastSimCO2 method was used to test 13 demographic models, including both population size and connectivity change in Microcebius arnoldi, by comparing the likelihood obtained from the model simulations with one obtained from the genomic data. The PSMC suggests a similar demographic trend for the two forest sites. The horizontal axis represents the time before the present, and the vertical axis the effective population size. The top horizontal bar identifies the 5 main periods of vegetation change according to the paleo-environmental reconstructions. Assuming panmixia, the PSMC inferred a population expansion during the African humid period that is in line with the expansion of the evergreen humid forest during this time. The Microcebius arnoldi largest population size also coincided with the maximum humidity observed in Mountain Dambre during the past 25 kiloyears. Finally, the Microcebius arnoldi underwent a population decline after the African humid period termination that coincided with increasing acidification in the region. Assuming population structure, The model parameters that better approximates the ICR inferred by the PSMC, represented by the blue curve, and the ICR inferred by the structure model, represented by the green curve, for Fantany, suggest a fourth change in population connectivity. The top horizontal bar represents the connectivity change, where warmer colors correspond to higher connectivity levels. Our simulations suggested higher population connectivity during the less glacial maximum and lower connectivity during the African humid period. However, such results are not in congruence with the paleo-environmental reconstructions, and this model was consequently rejected. The best demographic model revealed by the FastSimCo tool suggested two recent population declines. The colors in the figure represent different populations, and the arrows represent inflow. This model suggested the existence of a large panmetic population that became structured and reduced in size about 5 kiloyears before present, which coincided with the African humid period termination. This decline was then followed by a more recent population bottleneck during the last millennium, which coincided with the vegetation shift to grassland and the fire occurrence in the region. In conclusion, the dynamics of Macrocibus arnoldi could be entirely explained by subsection of population size change. This suggests that the expansion and contraction of the evergreen forest played an important role in demographic dynamics of this species. The old demographic dynamics were exclusively shaped by natural climatic cycles, while recent dynamics were likely shaped by both climatic and anthropogenic change. Altogether, our multidisciplinary study suggests that population dynamics were strongly impacted by climatic change in northern Madagascar during the mid-Holocene, before the human impact started. The next step of our project consists in recording the environmental and demographic change in the lowland areas. Thank you very much for the opportunity to present our project in this workshop, and also for your attention.